Welcome to the show, Aruna. Nice to have you.
Yeah, thank you, Guru, for inviting me to have this chat with you.
Perfect. Aruna, do you want to start with a quick introduction about you, your company?
Sure, yeah. So about myself, in terms of my academic background, I have a undergrad in biochemistry from UBC, and then also a PhD from UBC as well in cancer biology. And then I ended up starting this company during my last year of my PhD while writing my thesis. Me and a couple of my friends, we co-founded this. And then a number of years later, with that experience as well, I went on to actually got my MBA as well from Simon Fraser University on the technology program. Kind of a little bit of my academic background in a nutshell.
That's perfect. Do you want to talk about your scientific journey before we talk about your entrepreneurial journey? Like how you ended up here as the CEO of Vaxit, how your science experience even helped start your scientific journey. So should we talk about your scientific journey, like how you become a scientist and then an entrepreneur?
Yeah, personally, I became a scientist from a young age. I was quite curious. In the early 90s, it was the time of the late 80s. Molecular biology, genetic engineering was becoming a major area. It's a cool area. My dad was also a scientist, but he was a scientist in soil chemistry. So he used to get a lot of magazines regarding, plant genetics and so forth. So I was kind of curious even in my young age. I was flipping pages and I was quite interested in molecular biology and genetics at that time. And so then, young age, high school days, I decided that I want to do a degree in biochemistry and continue with that through that curiosity. So after high school, I applied to UBC, and actually I grew up in Sri Lanka. I was born in Canada, Saskatoon, and moved to Sri Lanka at a young age. I applied to UBC, came to UBC, did my undergrad in biochemistry, and I was fortunate enough to get introduced to a new PI who was setting up his lab at UBC in my during my third year. So he invited me to come and work in his lab to help to set up the lab. So I worked in his lab, and when I was finishing my undergrad, he asked what I'm going to do. And I said, definitely, I want to go to grad school. So he asked, why don't you come and do grad studies in my lab? So that's where I started my grad school. And so I worked with him for a number of years. And when I was finishing my PhD that last year, interestingly, another friend of mine who was in Oregon at that time, at the Oregon State University, contacted me and asked whether we know if anywhere can do some plant histology. So they wanted to get some plant specimens processed for histology and get in some sections. I looked around and I knew these two guys who were running histology, not a company, but they were doing like part-time work on the side, two histotechs. And I approached them, and their company name was Vaxit. And I approached them, and at that point, they had actually decided to stop doing that. So then we looked around, and there was no private facility that we knew who could do this. Mainly in the hospital lab, a lot of the research work also end up in the hospital pathology lab. And then what happens is, they get lower in the priority and it takes a long time to get things done. So we saw the opportunity. And then we also knew that Vaxit name was kind of known in the local community at that time because of that part-time thing they were doing. So we approached them and looked at ways to buy them out. And so they had shut down everything at that time, so they didn't have much. But we purchased the name from them. And me and two of my colleagues who were also grad students at that time. And so then we started to set up a lab. And our department head at that time in the Department of Anatomy and Cell Biology at UBC. He was very supportive, helped us to use one of the labs, the histology labs in the department to get started as long as we don't interfere with the department work.
Very cool.
That's how we get started, yeah.
And what were the main barriers you faced while starting the company. So I know in other industry, if you want to start a company, you don't need too many resources. I mean, it's always difficult to start a company, but especially in the biotech related work, it is capital intensive. So do you want to talk about the barriers you faced, or what was your struggle while setting up your shop?
Yeah, so at the beginning, financing was one of the major barriers, especially given that three of us who were starting this, all grad students, everybody had student loans and other debt at the time as a grad student. It was quite difficult. We went to a number of banks and they all turned us down. Except CIBC at that time gave us a line of credit, a small line of credit. And then three of us, we collected everything we could collect personally. And then we also knew, because we're setting up a service business. So we knew we can establish cash flow and then grow that way. So that was the advantage. We were looking at a way to generate a revenue stream to establish ourselves.
Right.
So that was the main challenge was financing. Yeah.
And financing, like equipments, space, hiring people.
Yeah. So equipment was a challenge, space was a challenge, but luckily we had good supportive faculty who helped. UBC is quite entrepreneurial as well to begin with.
Yeah. And did you get to use any university resources while setting?
So our department head at that time actually allowed us to use one of the teaching labs that they were using for medical school for histology.
That's pretty cool.
The understanding was, as long as we don't interfere with the department work or teaching work, they gave us the support. So, we ended up doing a lot of late nights. As a startup, three of us, we did from doing the work to doing the books to cleaning the floors.
Right.
We did all that.
I'm going through that right now. Yeah.
It's a big commitment when you're starting. And when you look at entrepreneurship, doesn't need to be just biotech, but in any industry, people have great ideas.
Right.
But one of the biggest barriers is the scared of failure.
You're right.
The people do not want to start things with wondering what will happen if I fail.
Right. Right.
So you could have a mediocre idea, but you can take it very far if you're not scared of failing and learning through those failures. So
that's true. I started my company about 6 months ago now. It feels like I fail like 10 times every day.
Oh yeah.
Right. So I receive rejections from investors. I receive rejection from potential customers, from potential partners. Some vendors don't take us seriously because we are too early.
Yes. Yeah.
It's a pretty uncomfortable journey, but I think it really makes you stronger, like as a person.
Yeah. Yeah. It pays off in the long run.
Yeah. Yeah. So it is not just you end up creating something that impacts the world, but also you become a better person. You learn how to be a stronger person and how to face challenges. So I think it's pretty rewarding.
Definitely. Yeah.
Yeah. Cool. So what advice would you give to someone who wanna start a company like Vaxxed, service-based, let's say a CRO or other related company? How is it different compared to when you started? And what advice would you give based on your learnings so far?
So, yeah, there's a lot more competition now than when we were starting. But I think, even with competition, if you find a niche area that you can support, and if you can better differentiate yourself from what's out there, and, jumping with both feet,
right?
It works. It
right.
works. Right. So even if you start and if you fail, you'll be able to find out why you fail and to pivot and still make it work.
Right.
So I think for the new upcoming entrepreneurs, I would say go for it.
Right. Well, that's pretty cool. And in terms of resources and a barrier to entry, I think it's much easier to start a company? I know there's more competition, but do you think it's much easier compared to your time?
I think it's much easier. People who invest anywhere from friends and family all the way to institutional investors, they're more savvy now. There's
Right.
a lot more money, a lot more interest. And actually one of the silver linings of the pandemic is that the biotech life sciences had gotten a heightened interest by the investor. You see news, see every day. There are large and large investments in biotech and pharma,
Yes.
and a lot more new companies. Actually, I think there are a lot more companies have started during the last 6 months.
Right. Right.
So yeah, and it's also in the public domain now. So everybody knows the importance of healthcare,
Yeah.
importance of drugs and vaccines, and so people are willing to invest now.
You're right. It became like the mainstream industry,
Yeah,
like a healthcare, biotech. It was always considered as like a niche industry.
yeah,
But now it is like everyone is getting attention towards that. So
yeah. And
this,
also when you look at all the big pharma that actually came up with the vaccines, the Pfizer or AstraZeneca, J&J, Lilly, the technology actually came from our little companies.
that's
They
true.
took it further, right?
That's true.
So when you look at like Biotech and Acuitas here at UBC,
Right.
they are the ones who excel the Pfizer vaccine. When you look at the Lilly drug, Abcelera locally in Vancouver are the ones who identify the antibody. So a lot of the smaller biotechs are making a big impact now. And it's getting recognized and getting a good payoff as well.
You're right. I was reading somewhere the return on research investment when big pharma does the research is like 1.3%.
Yeah,
But when smaller companies does that, it is over 9% return on research
yeah,
investment. So
yeah,
it doesn't make sense for bigger companies to even worry about innovating or creating. I think they will play a great role as a support entity and as
yeah,
a VC, as a venture capitalist, rather than as like a doer. I think smaller companies will lead this biotech revolution. I have full
yeah,
confidence.
definitely.
So this is a great segue to your company. So you talked
Hmm.
about the challenges you faced, how
Yeah.
you started the company. But what is it, and why did you end up creating this company? Like, what was the problem you identified, and why Vaxit? Why not something else?
Yeah, so at that time we saw the need. When we actually looked at, Western Canada, there was not a single private histopathology company. So we saw the need, and we saw we can create a demand as well. So for any, just to take one step back and explain what we do.
Mhm.
Um, So what a histopathology company does is, as a CRO, any drug development, vaccine development, any medical device development, part of that is like testing in animals, testing for efficacy, testing for toxicity and all that. And so one of the last step is in an animal trial is to look at what the pathological effects are. Is the treatment working, the device working? Are there any side effects? Are there any toxic effects? Are there any local inflammatory responses? Things like that. So one other way to look at them is looking at the tissue environment. So if somebody asks me what the histology lab does is, the easiest way I explain is, say you go to the doctor and the doctor says that you have some abnormal growth and we need to do a biopsy.
Mhm.
So they would actually do surgically or using a needle aspiration, take a piece of tissue
Mhm.
and they send to the hospital lab
Mhm.
and they will process the tissue, prepare the slides, stain them, and the pathologist will look under the microscope and make a diagnosis whether it's normal tissue, there's nothing wrong, or it looks like it's a tumor or cancerous and so forth. So that's in the hospital lab. That's what we do for drug development.
Mhm.
After animal trial, they would sacrifice the animals and collect all the tissues and send to a company like us.
Got it.
We would process tissues, prepare those slides, and we would do any specific staining that need to be done. And then we are working with a number of pathologists, read the slides and report on that study. So we do a wide range of things like that. So under histopathology umbrella, we do anything related to working with tissues, implanted devices, and so forth. So we do not have an animal facility. So the way we work is our clients would work on their own animal facilities. So some pharma, big biotech would have their own vivariums, animal facilities in-house.
Right.
They would do the animal trial. At the end, they will do the necropsies, collect the tissues, and ship to us. Or it could be a scenario where they are working with another CRO who does the in-life animal part. So they are specialized to doing the animal work. At the end, they will sacrifice the animals, do the necropsies, fix the tissues, and ship to us.
Got it. So do you collaborate with CROs directly, or you work with their sponsors, their customers?
All the way around. So we support all the way from early stage discovery to governments to all the way to big pharma. So we could be working directly with a CRO where they will be working on the sponsor studies,
Right.
or we could be working with the sponsor and they will run the animal study at a CRO, different CRO, and send us for the histopath.
Great. And so you shared one application where you can add value. You talked about when they do the safety testing or efficacy testing, or when they want to see the impact of the drug. So they send you the sample to analyze the sample.
Yeah.
Is there any other application? Do you want to give what type of requests you receive and from how many industries? I think you mentioned medical device, pharma, biotech. Is there any like a unique type of request you receive or other industries too?
So we do some government work. So there could be government studies. We have been working with, Ministry of Fisheries and Environment.
Yeah.
So we look at, fish samples, for different applications they do. Or could be toxic spill and they wanted to look at the impact on the wildlife, say fish.
Got it.
So say for fish reproduction, can collect gonads and other tissue organs and ship to us to do the same. So we do those kind of things. We also do plant histology, which is a very unique area because it's very difficult to do process plant tissues because of the cell walls. It's very hard to exchange the water in a plant tissue and infiltrate with wax for sectioning. So we have developed custom methods, custom protocols to do. So we have worked on a number of government projects and academic projects on plant histology. We do a lot of medical device work. So we have a very unique niche area that we work on, implantable medical devices. So there are certain types of medical devices and they're actually getting more and more complex now.
Right.
A medical device is not a single material anymore. You could have metal, you could have different plastic components, you could have different fibers.
Right.
So one of the problems doing histology on an implantable device is that, so you have a implanted device, let's say conversation purpose, so let's say a stent. A stent could be made out of metal.
Right.
And now they also have drug-coated, drug-eluting stents as well.
Right.
So when you implant a stent, now you wanted to look at, whether there's any scar tissue formation. If you implant a stent and if there's a scar tissue formation and blocking the artery, there's no point of doing that.
Right.
Or you can get a very high inflammatory response because of the stent material. Could be get a response because of the drugs that it's eluting. So because of that, pathology is a requirement to look at like local microenvironment where the stent is implanted. So say in an implant study, say you implant a stent in say a pig model, and you would collect that piece out. So now with the tissue around it and you have a stent embedded, if you process into paraffin sectioning, so you process into paraffin, and if you go to section, when the metal hits the blade, it will, shear through the tissue.
Mm-hmm.
So you cannot really section implantable material in standard paraffin processing. So we actually have developed methods to embed everything in a resin.
Right.
So using the plastic, we can control the different plastic types, and we can control the density of that.
Mm-hmm.
So what we can actually do is then after that, if it's a soft material, it's a soft fiber type, people use it for ACL damage repair, so and so forth. The plastic, or it could be fibrous scaffolding can use.
Mm-hmm.
Same with for breast implants as well.
Mm-hmm.
Those ones we can embed in a soft plastic, and we can section it at 5-micron thin sections. And then we can put them on the glass slide, and then we can remove the plastic and stain like a regular tissue. That way you actually maintain in the device tissue interactions.
Right, right.
And actually get a really nice readout on the local microenvironment on that.
That's very cool. So this test is more suitable if the device is targeted and localized. But these days there are many devices. I don't know if you would call ingestible capsule a medical device. It is a device in a way, right? So it can be targeted to one area, but sometimes it is moving to different places. Yeah. Have you received any request?
We have done work where you look at different areas of the intestine.
Right.
To look at local changes. But going back to the other type of devices, something that has metal.
Right.
What we can do is actually we have methods developed to put it in a plastic. Mm-hmm. And then what we do is we, using a diamond saw, we cut the first surface, mm-hmm. and we can polish that surface. And then you attach that to a slide, and then we can do a second cut. So now you'll have about a 200 micron thick plastic section with the metal containing device.
Right.
And then we can grind and polish it all the way down to like 10 microns.
Got it.
And then you can do staining. So again, with like very hard devices or even bone, we have developed methods to look at the histopath of the device. So you can like looking at biocompatibility issues, looking at toxic issues, inflammatory responses. You can look at real time on the device while the device is physically interacting with the tissues and integrating into tissue. Right. And the standard way of doing this has been, there are fairly expensive equipment to do that. But again, those ones are about, when you section, their sectioning is about 50 to 100 microns thick. So we have actually increased the resolution by being able to bring it all the way down to 10 micron thin sections. So now you can actually have much better resolution under microscopy to get a much cleaner readout on what's happening around the tissue.
That's pretty cool. And
so that's another unique area. So anywhere from, wildlife or fisheries or plants or implantable medical devices, and then to regular standard histopath. And then immunohistochemistry is another area we do a lot of work on. We do a lot of antibody testing and validating for, say, commercially developed antibodies. So if a commercial vendor wants to look at applications for immunohistochemistry, so we can develop biopsy protocols for that. With our biotech and pharma clients, wide range, sometimes we get a request saying that we are interested in this particular protein target. Can you help us? So we'll do all the background research, identify commercially available antibodies, bring them in, develop IHC protocols, and then we end up doing the study.
Got it.
Then there are ones, biomarker discovery type work, and then companion diagnostics. So you're treating with a certain drug, and you wanted to get a readout using IHC. So we have developed a companion IHC assay to do a readout. We have worked on studies for patient selection for clinical trials, because if you pick the right patients for your trial, you have better outcome on the study. Yeah. So for example, there's this one study we did for a pharma client where we identified 14 different targets in the signaling pathway, and we developed IHC protocols for all those 14 targets. And then we did a screen of available the tumor model, using the same type of tumors that are available from tissue banks. We screened and we found that 8 of those target proteins are upregulated or downregulated in this disease indication.
Right.
So we developed an assay with those 8 targets. So now they would go out when they're selecting patients, they do a biopsy, screen for those 8 markers, and depending on the expression profile, you decide that patient is going to go on to a trial or not. That increases the success rate on the trial because you're now targeting the kind of patients or kind of population that will respond to those drugs. So we use IHC methods to do those kind of assays development as well. Another thing is about the company. Even though we are a CRO, we actually work in a different model. So we do a lot of R&D work. So it's not the standard cookie cutter lab that you send your samples and you get a report. Where we work with the model where lab down the hallway kind of a model. So we want to be part of your research team kind of.
Right.
So for every project we do, we actually have somebody directly working with the research team, giving updates, giving feedback, having discussions that allow our customers to understand their project along the time and try to make the modification and changes they need to do so that they achieve their research goals. So answer the question they're looking to answer.
Right. So it is more collaborative instead of fee for service. I mean,
yeah,
it is for service, but with more built-in collaborative nature.
Exactly, yes. I think it's also, my background as well and my other former partners' background as well. We came from academia. At the beginning, we make a commitment to support academia as well. So we still support academic labs at UBC and other universities as well. But we also had that research curiosity. My team is quite curious about any, science projects as well. So we like to get involved and we like to have conversations with our customers and try to understand what they're trying to do, what questions they're trying to answer, and then help them to navigate that. So there are certain projects that straight up, they say, we want this and this and this done. Right? But then there are other projects that, we need help to navigate this. We need help to design this study. We need help to understand the data. So we work in a fairly collaborative way rather than being that black box where you would send your samples and you wait for your slides back or your images back or your reports back. So.
Very cool. There are a couple of things you mentioned. So the whole time I was thinking, uniqueness, I understand. You cover broad range of areas, different type of samples. You receive clinical sample, preclinical, and even from ag science or like plant sample or environmental impact of something like a fish sample. So is there any IP? Do you have any patent or intellectual property either on process or technology that you like?
No, so we do not hold IP. So it's a lot of know-how. So we maintain our internal know-how.
Right.
And we develop a lot of methods internally. So we hold them. But if we do not take on any of our customers' IP, so we actually return everything back to them. So that makes it easier. Any customer who wants to sign a non-disclosure agreements, NDAs, we do that up front so that they protect and they maintain their IP, their knowledge. And when a customer pays for a method development for them, that becomes their information. Yes. So, and then we also, as I mentioned earlier, we are also GLP accredited. So we get inspected by SCC, Standard Council of Canada, every 2 years. So any GLP study we run, they'll audit and do those inspections routinely.
Very cool. Very cool. And one thing you mentioned is you have your own R&D, internal R&D programs. You have your own products as well, like assays or all the R&D work you're doing, have you created any off-the-shelf products in addition to services?
Yeah, we are working on a number of our own internal products,
Mm-hmm.
reagents per se.
Mm-hmm.
But a lot of the R&D investments we have made is to develop processes to provide a better service, a better technique, a better methodology for our clients. So, for example, the implantable medical device histology, we made investments to develop methods. We made investments to modify equipment to be able to do that. Yeah, so it's purely for providing a better service, providing better assays to our customers.
That's pretty cool because like it's not just so industry is also changing, right? You have new
Yeah,
type of medical devices, you have new type of biology,
yeah,
and they have different type of impact.
yeah,
Uh, there's a new type of samples. Uh,
yeah. So we need to be able to pivot. So we actually have a really good system with the medical devices, implantable devices, with the understanding of the different components in most of the new devices.
right,
What we do is, any new device project we do, we actually do a lot of the pre-testing because when you're implanting, when you're processing a device with a resin material, we need to pick the right resin material too, because if that particular resin we use or the process we use could damage the device,
right,
then the outcome is quite different, right? So we wanted to make sure that the material or reagents we have to use have no physical impact on the device. So we do a lot of the pilot work with our customers before we actually do the actual studies. So that's another advantage customers have or collaborators have, because we always think about what they would want, what the outcome on their end. So we wanted to initially do a lot of the pilot work with them so that they have confidence in the results.
right,
So that's why that collaborative approach helps, because we actually, any testing work we do, we share that information with the customer and evaluate, this is this the right path to do? Is this the right way? Are you getting the right information you're looking for? And are you comfortable and are you happy? Right?
very cool. And why would any biotech or pharma company choose Vaxit over other histology? You mentioned many couple of uniqueness, but I want to understand like when customers receive, let's say, competitive bids,
Mm-hmm.
right, and there are many core labs in this space now as well.
Yeah.
So like, how do you like stand out? How do you outcompete?
Yeah, so right now, so far it has been based on that collaborative approach.
Okay.
Uh, because most of, pretty much all the customers we work with, they like that feedback loop, collaborative approach. Uh, then, we do not take projects for the monetary value per se. We wanted to make sure that they are getting best value. So
Mhm.
if something's not going to work, we tell them straight up, we can do this, we can charge for the project, but I can tell you that you're not going to get the outcome. So, you know, don't waste your money. Let's look at it in a different way. Uh, so the driving force is not just money. We wanted to actually uh, have some impact in science,
Mhm.
impact in R&D. So often I have told customers that, I don't want to, charge you for this because I know it will not work.
Right, right,
So, the customer's happy to know that they can actually, then they will ask for help to redesign the study or look at it in a different way, looking at a different markers for, say, it's an IHC project, let's look at different targets and so forth. So why would somebody come to us? I think a lot of the time it's that unique approach. And we are not like a standard CRO. We do a lot of R&D work for our customers. They like that as well. I would say, for new customers in a new type of project, we always say rather than jumping into the big study, do you want to do a pilot? just to get an understanding how we work with you, and are you getting the best value for your study. And if we are confident, then we can move forward. So
Make it mine. I think collaboration and trust.
that goes a long way, yes.
Yeah, there are now, not in histology particularly, but there are over 15,000 CROs now.
Yeah,
Every domain, you have so many different options. So there's
yeah,
decision. How would you evaluate them? Right. So
yeah,
most companies look for somebody who has done this before
yeah,
quite a long time. There are people who are happy with the outcome,
yeah,
and also they can rely on them and work collaboratively, because it's not just like, it's not a factory, right? So you
yeah,
cannot just tell, hey, that's need to be produced, do that. It is
yeah,
more collaboration. Even when the work is outsourced, you're still figuring out things, right? Upfront, you always have scope, but still there are many things that you learn while doing it. So it
yeah.
does, so it is
Yeah,
very
and if it's something that we haven't done or if we can't do, we'll tell upfront,
right,
saying that, okay, we haven't done this, but if you are willing to work with us and we are willing to work with you to develop the method that would work for you.
right, right,
So, so that works quite well for most of our customers.
right. It does make sense, especially with the pilot approach. If
Yeah,
something is unique, I've never done it, it makes sense to run a small POC or pilot,
yeah,
figure out how you can even scale that program.
exactly.
Mhm. That's pretty cool. And so, like, industry is changing. You mentioned you already innovated quite a bit in last decade or so. So where do you see this industry is going, and what type of disruption, what type of innovation you will have to make internally to really stay at the top of everything and be competitive this industry?
So in our way, I think the sky is the limit right now for the industry.
Right,
Especially right now, as I mentioned earlier, after the pandemic, everybody is so upbeat about biotech and pharma.
right,
Uh, People in every corner of the planet had suddenly realized the value that biotech and pharma creates.
right,
You know, who would have thought we'll be able to have a mRNA vaccine within less than 12 months? You know, it's remarkable, right? So people suddenly realized that, this is not like a side industry. This is vital to our survival.
right,
And also always people used to complain how expensive drugs are.
right,
But when you really think of it, a pharma company will start with like 100 candidates, and only 10% of that will make into the clinical trials,
right,
and only one of that will actually come out as a drug. So that means you actually need to have investment for the other 99 candidates that failed. So that one drug will carry the cost of that 99 candidates. So
right,
that's why it's so expensive, right? So investors are not investing, will not be able to create new drugs. So I think the right now with the renewed interest
mhm,
globally and seeing a lot of investment going on, I think there is a lot of opportunities. There's a lot more drugs, especially with now knowing how mRNA vaccines work. The number of applications are unbelievable.
right,
You could have personalized treatment for many of the cancers in the future. Right?
right,
And then there are a lot of other diseases that we are not looking at now. So I think for us, there is a lot of opportunity because, all these drugs or devices need to be tested for efficacy, tested for toxicity. So in terms of pathology, the demand is growing. As I mentioned earlier, there are complex implants, implantable devices, implantable materials being developed. So all those need like unique ways to look at
right,
biocompatibility. So we are constantly developing methods. We are looking at the future and trying to develop methods how to do that. Um, Immunohistochemistry, target identification, biomarker discovery is growing. So there's a lot of demand there. So I think there is enough work for a lot of the CROs to continue to grow in this area.
you're right. And I agree with you that it is really remarkable that we have a vaccine within a year almost.
Yeah, exactly.
Clearly boosted some confidence. It shouldn't be a norm to bring one drug or vaccine to market and take a decade or 15 years, right?
Yeah.
It should be a norm.
No,
If we can,
exactly.
if we can do it for COVID, I'm sure we can do
You
it
can do
for
it for other things as well, right?
diseases that are waiting for cures and treatment desperately.
Yeah.
And we should take advantage of that confidence and really declare a global urgency that we want to have disease-free world and maybe reprioritize our focus from life saving to life enhancing once we have all the cures. But
Yeah.
it does require global leadership. It cannot be done by US alone or Canada alone. Global collaboration like we have seen in the COVID.
Yeah.
So, like a German startup collaborated with US-based big pharma.
Yeah.
Now leveraging India for manufacturing quite a bit.
Yeah.
So, so it's like it was a global effort. Like Europe,
Oh, yeah, exactly.
China has their own vaccine. So
Yeah.
I think
And
we
actually, it's interesting, it's not really spoken much or given credit much.
right.
Local company Acutus,
Hmm.
actually across the street from us, they are
Hmm.
the ones who their delivery system
Right.
that the Pfizer BioNTech vaccine is using, their lipid delivery system.
I didn't know that.
Yeah, so it's a local company, right? So they develop the delivery system, and they have been working with, I think they have been working with the BioNTech before on other delivery mechanisms,
Right.
and that's what it is in the Pfizer BioNTech vaccine.
Yeah.
Their
Yeah.
lipid delivery system is what's been used.
Right. Right.
And you heard of Abcelera,
Oh yeah.
the biggest IPO in Canada. It's another little local company. They identify the first antibody for Lilly antibody treatment
Right.
within 6 weeks of the first COVID case here.
Yeah.
So
Epsilon is doing great work.
yeah,
So how is biotech scene in first in Vancouver and British Columbia and Canada, according to you? Do you think it's growing? Can we create our own greater Boston area in Canada?
I think so. I think so because right now, there is so much investment. It's you hear this large scale investments suddenly happening in the last 3-4 months, and locally, yes, Abcelera is growing huge. Precision nanosystems
Right.
growing huge. Taking leaps, and then a lot of other companies. Notch
Hmm.
Pharmaceuticals raised about 85 million about a month ago.
Right.
Uh, so there are a lot of interest and a lot of investments and a lot of growth. Yeah,
Yeah, I see
and there are a
that.
lot of jobs being created.
That's true, and I see this in greater Toronto area too.
Hmm.
Level of investment, interest, entrepreneurship. Moved here from Europe, and I
Yeah,
can see the difference. Like people are more entrepreneurial, VC and angels are more engaged. So I would be surprised if we cannot create the own Silicon Valley, own greater Boston area for
yeah,
because to get access to a bench space or lab in Boston, it's crazy expensive.
yeah.
Here it is very competitive. You have access to talent from universities. Yeah, I won't be surprised if we
And
see.
I think Canadian universities has always been very encouraging for company creation. So I know,
Yeah,
especially UBC. Throughout the history, the time I have been here, I have seen, so much interest or so much support from the university to start new companies. So
very cool. Aruna, it was a pleasure, fun talking to you.
yeah, definitely, it was a pleasure chatting. And
I will be in touch very soon.
okay,
Thank you, Aruna. Sounds
okay,
good.
take care.